Hi everyone, thank you for joining us. We will wait just a few more minutes to let everyone join us and we'll start. Good afternoon and welcome to NRGene's webinar series. My name is Dafna and I'm happy to be your host today. Today we'll discuss CannSCAN, our new efficient and affordable genotyping solution specially developed for hemp and cannabis. We'll save time for questions at the end of this webinar, and we encourage you to type in your questions during the presentation using the question tab on your screen. This webinar is also recorded and will be posted on our website if you want to listen to it once again. It's my pleasure to present today's speaker. Many of you are already familiar with Dr. Uri Weishaus. Uri holds a PhD in plant genetics from the Hebrew University of Jerusalem. He's been with NRGene since 2011, applying genomic tools in breeding projects for various crops. In the past several years, he has specialized in cannabis genetics and become an expert in the field. Uri, the stage is yours.
Hello, everyone. Good morning and good afternoon, and thank you for joining us in our webinar series for cannabis and hemp genomics. Some of you have already heard my talks on various topics related to cannabis and hemp genomics, and in today's session, I would like to tell you about CannSCAN, our new genotyping platform for cannabis and hemp. And I'll start with saying what you can expect from this webinar today. Okay, so I'll start with an introduction on cannabis genomics, followed by why we created CannSCAN, followed by what is CannSCAN and what's the purpose and what is it used for, and I'll show some examples of uses for CannSCAN. And then we'll have time for questions. So a quick word of the presentation that I will show is aiming for a mixed crowd. We know that not all of you are bioinformaticians or breeders or plant biologists, so we try to aim for a wide audience. And for some of you, it may be over technical, and for others, too technical. So please save your questions. You could either send them in the Q&A part or send emails following the webinar with specific questions. So a quick word of introduction about NRGene with those of you who are not familiar with us. NRGene is not a cannabis company. We are a seed company. We're not a seed company. We're a genomics company working for seed producers mostly, but also end products. We are located in Israel, the R&D unit, and most of the people are in Israel, and we have units in North America, the US and Canada. In the cannabis and hemp space, we work mainly with Canadian large licensed producers, but also companies in the US in the hemp and cannabis space, and also with a large company in Switzerland and in Israel. Our technology has been validated by over 25 top-tier scientific publications and used by industry partners, so we know the technology is highly reliable and verified both by market and academia. So a quick word about CannSCAN before I go into the details. CannSCAN is meant to be a first step in a array of services and products that we offer cannabis breeding. It can be a standalone, meaning you could use CannSCAN to create a genotyping pattern for your germplasm, but we really would like and prefer that it is a first step and you move from CannSCAN and understanding your genetic diversity and move towards the breeding program and do larger projects with us in terms of trait mapping and breeding and reaching a roadmap, and finally new cultivars developed with our genomic tools. So as I mentioned, the first and high priority step in any breeding program is to understand what kind of germplasm you have. So it's kind of trivial to cannabis breeders that you have to grow your varieties out and test them and do pheno hunting, or at least phenotyping in various degrees of thoroughness. But it's also crucial to understand the genetic makeup and the composition of your population. So the first step to do is to analyze and understand the genetic makeup of the germplasm at your disposal, get rid of redundancies, and select individuals that are important as founder lines. Once you know your founder lines and created a unique identifier for each one, you could start by designing your crosses and creating segregating populations and mapping populations, and start looking for traits and use genetic markers to in the breeding process to develop new cultivars. So a quick introduction to those of you who are not plant geneticists, terminology. So specifically, I would like to kind of hold for a minute about inbred or homozygous lines versus F1 hybrids. And I like this example, classic textbook example from maize, in which the parental lines in seed production are inbred parents, meaning each chromosome has two identical copies. This is achieved through generations of inbreeding or shortcuts like double haploids. But once you have two parents, in this case B73 and MO17, they have two copies each of each chromosome. And when you make a cross, you have F1 seeds, true F1 hybrid seeds. Each of these hybrid seeds is identical, and they contain one chromosome from the male and female parents. And this enables seed companies to have a very successful business in selling these seeds, but also employs and utilizes a phenomenon called heterosis or hybrid figure, in which the F1 hybrid performs much better than both parents. In this case, in a classic textbook example, the F1 hybrid, both the plant and the fruit or the actual product is better performing than two of the parents. In cannabis, the biology is more complicated, so it's not easy and straightforward to talk about a true F1 hybrid for several reasons. First of all, cannabis is a dioecious plant, meaning it has both male and female flowers either on separate plants or on different plants. So you could have male and female plants with different flower types. The other complication is that a genetic female is not necessarily a female in the pollination, in the crossing process. You could have sex reversal and force females to create male type flowers and pollen, so they become, you could have X males or Y males, either called pollen donors or X males or Y males. So the complication in the actual breeding process. And also, cannabis, because of its nature, does not prefer to be inbred, meaning if you keep selfing a plant till you reach homozygosity, like you do in the maize examples I've shown, in many cases you reach inbreeding depression, meaning the plants grow very slowly, they're not viable, sometimes the pollen is not viable, sometimes the seeds are either very few seeds or not at all viable, do not germinate. So it's a very complex breeding scenario to deal with. So true F1 seeds are not easy to obtain like you do in maize. So the cannabis and hemp industry have found two solutions for this. One of these is very common in the cannabis industry, to use vegetative propagation, clonal selection, meaning you take a well-performing plant that you know and you're very satisfied with its performance, and you have cuttings and propagate, you grow roots and you grow leaves of the plant, so it's vegetatively propagated and all the genetic makeup of all the individuals is identical. So this is an advantage, but it's also a disadvantage. Once you have a pathogen infect your crop, all your plants are as susceptible to this new pathogen. The hemp industry, on the other side, has come up with a different solution. In this industry, it's common to use seeds for logistics reasons. So in order to use seeds, you can't have true F1 seeds. You have something close to true F1 seeds. You have semi-stable or semi-segregating seeds, meaning you cross two parents or two populations of parents, in which some of the regions in the genome have been stabilized or fixed. In this case, those blue rectangles, they represent a region that's identical in the two copies of the chromosome. So once you make the cross, all the plants will not be identical, but they'd be close to identical, and all of them would contain the region of interest. So if this region contains traits of interest, you have those in all the individuals. So it's kind of a simplified way to present the solution, but remember, not all the plants in the hemp field are identical, but they're, for all practical purposes, they perform as if they were true F1 seeds. And this is how most of the industry works right now. So this kind of dual way of operating creates several problems. One of them is that you have to manage vegetative cultivation and vegetative propagation. You have to have mother plants or mother stock plants for production and for routine backup of next batch and for the continuation of the process. You don't start from seeds, you keep on cloning and recloning your plants. So this really means that you have a room with your precious assets, the mother plants, maintained like this. Usually you have to have multiple copies of the entire crops, because if your mother plants get destroyed somehow by fire or something disastrous like this, you need to have a backup for this entire operation. Otherwise, you lose your clone and you cannot go back to your production pipeline. So what people do is keep multiple copies and also keep seeds. So the seeds are not identical to the mother parent, but they are they may be very close to the mother plant. So this is one way to keep some sort of a record of the actual plants and take very good care of your mother stocks. But this also has large impact on how the operation and the cultivation is maintained. So if you're doing vegetative propagation, there's a lot of manual work, and this is a biological product that is very hard to label. The label is external to the plant. And either you're working completely manually or semi-manually or using even robots for the cuttings and propagation, mistakes happen. In an example like this, if this plant is removed from the tray, like on the right panel, it doesn't have a label because the label is on the tray. And if the label gets erased or lost somehow, there is no way you could come up with the genetic ID for this plant. And this is how mix-up happens. And if this happens on the mother stalks or in the early stages of propagation, you may have contamination, mix-ups, and wrong plants being grown in the right instead of the actual batch that you think you are growing. So the other aspect of keeping a breeding program and live collections is the germplasm collection. Most of the companies or the people engaged in cannabis breeding, especially in the medicinal or recreational space, they have a room like this where you keep all your collection of plants as clones, vegetative state. You need to keep multiple copies of this. This is not flowering at the same time. They're not susceptible and they don't like the same conditions. Some of them are tropical and like different lightings. This is a mess. This is labor intensive, space consuming, and extremely expensive to maintain. And it keeps expanding. Every cross you make creates new progeny. So you have a few individuals now that are added to your collection and you still need to keep more copies of it. So this is very expensive to maintain and you need a way to genotype and assess and get rid of redundancy. And then some of the redundancy, you could grow the plants and collect them and store them as seeds, but you still need and want to maintain the collection as small as possible. And then when you are collecting seeds and you have your collection in envelopes of seeds, which we like to call the mystery envelope, you have no real definition of what's out there because each seed is a snowflake. You have your heirloom collection, you have your legacy strains that have been purchased or obtained somehow, heterozygous siblings of certain crosses, either deliberate or spontaneous. You have mutations, also generated or intended. And then you have that bag of poorly labeled that says Purple Haze 164 with a cross that you're not really sure what it is, but you don't want to discard it because you think it might have excellent genomics. But on the other hand, you can't really grow all of them out and do expensive phenotyping. Also, you just keep the bag and wait for the right moment. So these are all problems with clonal propagation and reverting back to seeds without genetic ID of the makeup of your collection. So what is CannSCAN? In a very simplified schematic view, in CannSCAN, we analyze your cultivars. You send tissue samples or DNA samples to our labs or our providers. We extract the DNA and do our analysis on the genotyping panel. I'll go into the details of that soon. And we generate a unique genetic identifier per cultivar. Illustrated here is the barcode. It really is just like that. There's a pattern of 200 data points that are unique to each cultivar. So how do you go about creating this data set in a way that will be compact but representing of the enormous diversity in cannabis? So all of you who have grown a lot of varieties or a breeding program in cannabis know that this is a hyper diverse and highly heterozygous species, both in its performance of plant morphology or chemotypes, but also on the genetic level. So this is a dual trait again. On one hand, it makes the analysis complicated. The heterozygosity and the extreme diversity is difficult to analyze. But on the other hand, because there are a lot of differences and a lot of diversity, it means we could capture this diversity and convert that into a unique identifier for each individual plant. This is exactly what we do in our CannSCAN genotyping. So exactly that we did. We took a variant panel or created a variant panel that contains 46 de novo assembled lines, either from data that we have obtained extreme diversity coming from various cannabis and hemp types. We have generated sequencing data or taken publicly available sequencing raw reads and run all of that through our unique pipeline of genome assemblies and analyze all that enormous quantity of high resolution data and compared it all to one reference genome and extracted all the differences in this large data set. So as you can imagine, a lot of the regions are identical to the reference genome. Some are different from the reference genome, but are not biallelic, meaning they're different in some of the varieties, but they're more than 2 alleles. There are multiple versions. Some are small insertions or deletions, different color for each fraction, and a large fraction of the diversity is SNPs, SNPs, single nucleotide polymorphisms. They're not the only way to look at diversity, but they're a very easy and practical way to look at diversity. So this is what we're using in our genotyping platform. So to sum up the process till now, we have generated a variant panel with lots of genetic information or genetic differences between a very diverse panel, and 21 million of them, or about 21 million of them, are SNPs that we could use in the filtering process. So we went out to develop our genotyping platform. So we have the platform that we call CannSCAN. So we can't use all 21 million SNPs for various reasons, mainly because they're not useful. Not all of them are useful, and they're also, it's a large data set. We don't want a 21 million data point genotyping system. We want something compact and affordable. So we went out to filter this data set. So we started out with 21 million of the full panel SNPs. We filtered them into what we called a clean subset, which are valid SNPs that we have great confidence in their existence. They're biallelic, they're distant from other variants, and they actually place and map to the chromosome assemblies into known regions on the genome assemblies. So that filtered by orders of magnitude the data set into about 170,000 SNPs. We continued to filter the data set into reliable, meaning discovery based on discovery quality. I won't go into details of how and why we're doing this, but it's a rigorous process to also further filter the data set into SNPs that we can trust. And then there's a parameter called robust or robustness, which is the fraction of missing recall data, meaning every time you look at some of the data, you'll actually get a signal in a large portion of the varieties that you're analyzing. So that got rid of another 75% of the data, still a large number. And then we look at a parameter called informativeness, or we want informative SNPs. I'll explain that in a bit. That also considered allele frequency and to have non-redundancy in the data set. And finally, we want platform compatibility, meaning we want to have the suitability of the data set aligned with the way we're actually doing the physical assay, meaning GC content, complexity, ability to design primers, and things like that that are on the physical side of the assay. So once we have the 12,000 SNPs data set, we went out to do binning. We wanted even and good representation of the entire 10 chromosomes of cannabis, or 9 autosomes and sex chromosomes. We wanted full distribution of every region in the genome. We wanted to make sure we're representing that. So we went out and binned the 854 megabase per genome into 205 bins. So on average, and they're not identical, on average you have 4.2 megabase pairs per bin, and the entire genome is represented. And if you look at this example, and this is chromosome 10, there's ample number of SNPs for each bin. So each chromosome and each bin is represented with a large number of, or an ample number of SNPs that are both compatible, informative, and robust. So now we could go to the next stage and actually design a panel that will have all this performance and represent the entire genome. And we did exactly that. And this is the actual distribution on real coordinates, and the coordinates are public. This is CS10 genome coordinates. These are public coordinates. All 9 coordinates, all 9 chromosomes and the X or Y chromosomes, they're all represented evenly with some enrichment in regions of interest. They're all represented on the entire genome, so no region is ignored. And for having the data arranged in this way means you could also zoom in on specific regions and look at a subset of the panel. In this case, if you look at this region in chromosome X, and that all of these markers can distinguish between chromosome X and Y. So you could actually use this to also do genotyping and sex determination and discriminate between X and Y seedlings. But also you can choose other regions and do the same type of exercise in other regions of interest. You could choose a subset of the 200 and drill down and have that associated with the traits of interest. So now we get to the issue of informativity. So we want informative SNPs. We wanted a compact assay that has the number we reached was 200, because it's a trade-off between the usability and cost and the maximum amount of information and the accuracy and detection of multiple types of cannabis, hemp, and other and related cultivars. So we want informativity. So not all SNPs are as informative. If you look at the ones wrapped by a square, this T here versus the C's. So this T would help you discriminate between cultivar 12 and all the other. Same goes for this T. It is good for discriminating between cultivar 10 and all the others. but it's not the most informative SNPs. If you look at these, for example, this C could discriminate between these cultivars, but also number 2, and same goes for all the ones that are encircled in a circle. So we have taken these and the most informative ones based also on genetic considerations. We chose a subset of 200 individuals, and those create a pattern. So once we determine the most informative ones, we could collect them and create a pattern that is represented here as a barcode that is unique representation of each cultivar tested. So I'm gonna present now the uses and applications for this technology. And first of all, I'll show examples of each case. So the first one and most sought after and applicable application for this is to create a time-stamped genetic ID for each variety for registration and IP protection purposes. I don't want to go deep into how plant registration is done. It's different from country to country. I'll focus on the major cannabis growing countries. So in Canada and the US, in Canada there's something called PBR, Plant Breeders' Rights. In the US, there are various ways. The most important one for variety protection is called a PVP. You can register hemp plants in the PVP. You cannot register cannabis yet, but this is soon to change. You will be able to apply for PVP for cannabis as well. And then there's an organization called UPOV, that is an international treaty that does the same thing, plant registration worldwide. They all rely on the same principle called DUS, distinct, uniform, and stable. For our case, the most important parameters is distinctness. You have to show and prove that your variety is distinct from other varieties at the time of application. The best way to do this would be to provide a genetic profile that shows exactly that, and this is what CannSCAN is about. The next application for using CannSCAN is reduce the number of plants I've showed before in a collection. You want to save money. You don't want to keep a large ever-growing collection of germplasm. You want to keep copies, minimum number of copies of your plant, and minimize the rest. So imagine if you have hundreds of plants, either as clones or as seeds germinated for seedlings. Some of them are redundant. Some of them are genetically very similar to others. You need to have a way to genotype and analyze and cluster them. This is exactly what we do with CannSCAN. We provide the genetic analysis and relatedness of the samples that you send to us. We can tell you which ones are close to another within the batch that you send and which ones are identical and which ones are redundant and which ones are farther apart. So you need to keep copies. In this example, if your germplasm looks like this, and this is actual results of genotyping using our germplasm, using germplasm that we analyzed, the red dots in cultivar D, they're very close to each other. So you may consider to convert some of them into seeds and store them and not keep all of them as live plants. Whereas the blue dots are widely dispersed. They're genetically different from each other. So you may want to keep representatives from the extremes and minimize the collection in that way and convert all the rest to seeds. So this is a way to minimize based on scientific and genetic analysis, educated way to minimize the collection and minimize the cost spent on the breeding program. So the next to the uses and applications of CannSCAN is production pipeline. QC, quality control, either the mother stock mix-up that I mentioned can be identified in early stages and prevented. And you can have a low-cost quality control panel, either the entire panel or a subset of that, used for QC purposes, strain validation, make sure that batch-to-batch consistency exists. Detect counterfeit. People have told us that they have suspected samples that they think somebody's ripping off their brand. Is there a way to prove that? So yes, with CannSCAN, there's a way to show such counterfeit cases, you can test two samples and ask this question, are they identical or different? Supply chain validation, and then external validation. Seed certification agencies in other crops require genetic testing. In hemp, this is probably soon to also be accepted as a way to test authenticity and true to type. So an example of that, and this is a financially interesting case, look at only the red dots. The red dots represent a variety, let's call it the purple kush or variety A, whatever you want to call it. On average, it performs medium. And if the trait that we're looking at is yield, it performs medium. Why is it a medium performer? Because some of the plants are good producers, but some of the plants are poor producers. You think they're the same. They're all labeled purple kush or variety A, but some of them are actually got mixed up somewhere along the way, and they're different. They look the same, but you had no way to determine that. So if you do the genotyping QC, you could actually separate the subsets into poor performers and high performers based on genetic makeup, and have the high performers keep on growing them, and the low performers, poor performers, you could convert into seed and collect them for storage, but not grow them, and thereby increase your production efficiency. Another example of the uses is kind of a spin-off of CannSCAN. I'll show an illustration. So CannSCAN contains 200 SNPs and data points. Once we do the analysis and provide you with the data, you could use a small panel for a custom assay in-house or send it to us. We can increase and shrink the size of the panel. We can have a larger panel for higher resolution, but you could also choose a subset of to have a kind of a in-house assay for very, very simple purposes. What if you analyze your entire germplasm and found out that 200 points give you a crude assessment, and that's very good, but in order to really distinguish between the two lines you're producing, you only need 4 data points. So you can now take those data points and we could design a sub-assay out of that, and you could do an in-house, simple, cheap assay and maintain production QC. So that's another use. It's more complicated than the other trivial use, but we're also able to provide that. And the last, but I think the most important use of CannSCAN is a deep understanding of the diversity that allows you to design and optimize your breeding program. I'm going back to the first figure that I've shown. You need to understand the makeup of your germplasm, analyze cost effectively the first initial germplasm you're starting with, design your crosses and your breeding program based on genetic distances, take the most extreme cases, and in each group, select individuals that are farther apart on the genetic level and increase the likelihood of creating new combination in the crosses and in creating segregating population. So we think this is one of the most important uses of CannSCAN. So the take-home message that I'm leaving you with is this simplified graphic message that with CannSCAN, we are accepting your germplasm samples. You send us 200 or more samples of your germplasm. We analyze with our providers the genetic makeup, and we create a unique genetic ID for documentation and validation, time-stamped for your germplasm. And in the same time, you could use the same type of data in the breeding program and use it to advance in the breeding program and work with us on creating new cultivars. With that, thank you very much for listening, and I'll be happy to answer questions.
Thank you very much, Uri. I believe we have time to take a few questions from the audience. So let's start. Can you design a custom version for my breeding program?
Sorry, for what?
for my breeding program, can you design a custom version for my breeding program?
So the answer is...
Wait, I believe now people can hear you.
Thank you. So the answer is yes. The CannSCAN is a well-defined product. There's a standard genotyping panel. We can design a custom genotyping panel. The easiest way would be to do CannSCAN once and take a subset of CannSCAN and take the ones that are most suitable for your breeding program. And if you want to design a specific higher resolution panel based on our data set, we can also do that as a service, as a customized service. So feel free to reach out and talk to us for more details.
Thank you, Uri. We have another question. Is 200 SNPs sufficient to cover the entire genome?
Yes. So as we have shown in the one of the first slides, once we've binned the SNPs, we made sure that they represent each part of the genomes in all the genomes, and we've tested that and validated with real cannabis and hemp samples. So we know we are able to reconstruct populations and to use these exact 200 individuals for accurate representation of germplasm delivered to us. So in some cases, and there are cases where the resolution is not sufficient, but in most cases analyzed, this resolution would give you all the information you need. For example, if your genotyping missed one SNP in the panel of 200, so one of them will give you a no answer, you still have 199 SNPs to characterize your germplasm, which is way more than you need. People have done successful breeding programs and analyzed germplasm with as low as 20 or 100 SNPs. And we're talking here about 200 well-selected and carefully selected SNPs.
Thank you. We have another question. What are your terms if we work with you on breeding projects, create varieties? Will you take joint IP?
So the standard way that we operate, and in this case for CannSCAN as well, we work on a service-based. We do not have a claim or use your data for anything. We don't. We're not growing cannabis. We're not creating our own breeding program. We do this as a service. So that's our default way of operation, and we don't use customers' data for anything unless we have a written agreement to do so.
We'll take another question. Along the lines of the previous question of the SNP set, are you able to comment on the breadth of the genotypes of cannabis you used in developing your elite SNP set?
That's a very complicated question. We used as diverse as we could access, and I could tell you that because we work internationally and we work with companies that are distributed on various continents, we have had access to very diverse germplasm. How diverse? That's difficult to assess. We looked at the minor allele frequency and of the varieties, and we also got raw reads from public sources. So we tried to have the data as diverse as we can, and then compromise with, or at least reach a balance between highest resolution and most informative and cost effective. You're trying to kind of balance between three parameters here. Of course, you could have higher resolution panels. Those could be designed from the subset I showed. If the panel is bigger, and we'll need to work on the design, and also the genotyping itself will be more expensive. So we deliberately chose something that is compact and that would be cost effective.
We'll take another question. Can you give some information about the cost of the software and who owned the data?
So the first, I'll answer the second question first. You own the data. We do not own the data. We provide you with a genetic ID, a VCF file, and other forms of reporting, and we deliver that to you. And that is, it can and it is anonymous. We don't need to know what the sample name or even who the owner is, and we don't have any. As I said before, we don't use the data in our system. Once we are provided the analysis, it is deleted. In terms of cost, we are talking at significantly less than the existing solution. The existing solutions in the market are kind of an overkill in the fact that they provide high resolution genotyping, naturally more expensive. So we're talking about in the range of tens of dollars. It depends on volume. So we are analyzing 200 samples and more. If you're doing 1,000 samples, but in all the genotyping assays, the price goes down the more samples you perform.
Thank you, Uri. We'll take another one. How well does your panel differentiate full SIBs?
Differentiate what?
Full SIBs.
Oh, okay. So we are actually testing that very question right now. We have tested this with already with a small panel of full SIBs. We've been able to detect all of them. It depends on how homozygous the original germplasm for the full SIBs was, meaning if the full SIBs are more identical to each other because they started out more identical, it would be different. So far, the number of the plants we looked at, we've been able to detect all the full SIBs. We're looking at hundreds now, and we're just validating with higher numbers of full SIBs. I believe that maybe not all 200 SNPs would be useful in this case, but still you'd have a very large number of unique patterns for each one of the SIBs, so you'd be able to tell full SIBs apart. I'll actually have data to show in a few weeks with actual results from a large number of full SIB analysis.
Thank you. We'll take the last question. You show a number of triallelic SNPs. How common are these, and how confident are you that those 3 alleles are present?
Okay, so we can have more than when you mean the possibilities are 4 alleles, and you could have combinations of SNPs. We employed a rigorous process of filtering. I explained some of it in a really large overview, but this was a very large bioinformatic efforts by a large group of experts to actually filter, and you can see that we have zoomed in from 21 million that we started out with to 200. So you could be sure that the 200 that we chose, we have great confidence in them, otherwise they wouldn't be in the panel. That's kind of the broad answer without going into details again.
Thank you very much. I'm sorry we couldn't answer all your questions, but please note that we will follow up by email and answer all of them after the webinar. This webinar was recorded and we'll have it available in a few days, so keep an eye for an email from us with the link of the recording. You'll also be able to find it on our website. If you have any further questions, please don't hesitate to contact us at NRGene.com. Thank you for joining us today and enjoy the rest of your day.